sempre.
Okay.
Ho finito ieri mattina, quindi con molto tempo
So,
in anticipo. Come stai?
bene, tu?
Sì, bene, benissimo. Il tuo papà?
Sta.
Sì? Ok.
Okay.
I have some sound problems, I'm sorry.
Do you think we can start the session?
Hello, hello, hello.
Hi, Rumiana.
Hi, nice to see you, Roger.
Nice to see you.
Federico.
Despite only virtually.
Yeah,
The
yes.
pity.
Yes.
So do you think it's time we start the session?
Yeah, yes, it's 50. It's exactly 50.
Okay. So good afternoon, everyone, Tokyo time, and good morning, everyone. It's Europe time. So, and welcome to the last scientific session of this conference that is devoted to spectral pre-processing for aquaphotomics. So we will have three speakers, and it's a great pleasure for me to introduce the first speaker of today. That would be Dr. Jean-Michel Roger from INRAe in Montpellier. Jean-Michel is well known for his contribution in the fields of chemometrics, especially applied to NIR spectroscopy, in the fields of orthogonalization, variable selection, and of course in pre-processing that he was going to speak today. He received the Thomas Hirschfeld Award in 2016, if I remember well, and he created recently, what's like 2018, the ChemHouse project as a pole of research for an interaction between chemometrics and NIR spectroscopy. And today, Jean-Michel will speak about new trends in the pre-processing of near-infrared spectra. So Jean-Mi, please, you have 30 minutes including discussion, so try to stand within 25 minutes to allow some question and answer. So Jean-Mi, the floor is yours.
Okay, thank you, Federico. I will try to share my screen. I hope, can you see my screen?
Yes, we can.
Okay, thank you. So my name is Jean-Michel Roger, so I'm from France, and I will talk about new trends in pre-processing for near-infrared spectra, and I will try to focus on the aspect related to aquaphotomics. Okay, so the outline of my speech, I will start with an introduction, and I will focus on why to pre-process near spectra, and more, I will focus on some limits of pre-processing with two examples. And then I will talk about 2 points. The first is about the normalization procedure, which is a very, very often used process, pre-processing, which is very used in near-infrared spectroscopy. And I will finish with a point of view about how to combine pre-processing, and I will show that maybe it's better to fuse the pre-processing than choosing one or doing several pre-processing in a chain. And I will conclude with some take-home messages. So first of all, the theory, I think that everybody knows the Beer-Lambert law, but I just wanted to recall here that in the Beer-Lambert law, we have two things we can search about: the concentration of the sample and the extension coefficient, that is to say, the pure spectrum, in fact, of the sample. The Beer-Lambert law relates the absorption, which is the logarithms of the ratio between the two intensity, after and before, relates this absorption with the product of the extension coefficient, the concentration, and the length of the path length of the light inside the sample. So it's very simple. So near-infrared spectroscopy can serve two aims, in fact, two main aims. The first one is to retrieve the spectrum, the pure spectrum, the extension coefficient epsilon of lambda. And this is what you are doing, I think, really often in aquaphotomics, but we can do also that in forensics, arts, archaeology domains. and also in characterization of counterfeits in pharmacy, a lot of things. We are not interested in the concentration, but we are interested in retrieving the products, retrieving the spectra of the products which are inside the sample. And another aim which relates more which is more concerned in chemometrics is to calibrate spectrometers, multivariate calibration, and also to analyze the hyperspectral image when we are searching for the concentrations. So in theory, everything goes well because when we have a spectrum, we can say that the shape of the spectrum contains the extension coefficients that we can retrieve the extension from the shape of the spectrum, and the height of the peaks is related proportional to the concentration. So in theory, everything is okay. But in practice, I will take the example of light scattering. There is a lot of effects that can affect the absorption level and also the shape of the spectra. And here I have taken the example of light scattering, and I will keep this example of light scattering because it is the main effect we are searching to overcome in preprocessing. So you have in this example, we have 3 samples. This example is taken from a book of Phil Williams. It's a very classical example. You have 3 samples which are made up of exactly the same glass, okay? But so the chemistry which is behind these samples is exactly the same, but on the left, we have grown the sample in very, very finely. So we have a very, very small particle size. And in the middle, we have a medium size. And on the right part, we have very, very big particle size. And you can observe that the color, that is to say the absorption, the apparent absorption, the apparent spectrum, is completely different. You can see that on the left, the sample appears as white, and on the right, it appears as green, which is the true chemistry, the true color of the glass. And this is quite understandable because with a fine, very small particle size, the light is scattered. As soon as it enters the sample, it is back scattered. So there is no interaction between the true samples, between the small particle of glass and the light, because the light is retro-diffused, retro-scattered very, very rapidly. And despite on the course, the light can go through the grains and can be affected by the color, by the chemistry of the sample, okay? And so you can see the difference on the spectrum. Okay, so you can see that with exactly the same sample, with exactly the same chemistry, we have completely different spectra. So as a consequence, preprocessing is very, very important in near-infrared spectroscopy for both concentration estimation and extension spectral identification. So the first task, the first aim concerns more the chemometrics, and the second one concerns more the discipline like aquaphotomics. Okay, so let's come back to the Beer-Lambert law, and we can make an empirical model, which is often done in near-infrared spectrometry. We can state that the original absorbance is multiplied by a factor, which is called a multiplicative effect, and is added with a function of lambda, function of the wavelength, which is due to the loss of function of photons, and it is called the additive effect or baseline. So with this very empirical model, we can imagine 2 kinds of preprocessing. We can imagine a first class of preprocessing, which is dedicated to remove the multiplicative effect, and a second class of preprocessing, which is dedicated to remove the additive effect. Let's see 2 examples of this preprocessing. So first, before seeing these 2 examples, this slide is just for resuming, for summarizing the 2 classes of preprocessing. So First example, we will see the example of derivative. This preprocessing is very, very oftenly, it's a very, very often used in all the disciplines which use spectroscopy, aquaphotomics also, and also multivariate calibration and so on. These derivatives are used to remove the baselines, classically. And here we have an example. Let's imagine a very, very simple spectrum, which is made up of one peak. This peak is centered at 600 nanometers, is 20 nanometer wide at mid-8 here, okay? And this spectrum has been added with a baseline, a parabolic baseline, which is a very, very classical shape for near-infrared spectroscopy. So we have these two peaks, and we will try to remove the baseline using a derivative. So I have applied Savitzky-Golay, first derivative, in order to search for the maximum of the peaks. And because I have computed the first derivative, I am searching for the zero of the derivative. You can see here for the blue pure spectrum that the zero is at 600. Everything is okay. But for the red spectrum, which is the spectrum added with the baseline, we can see that the zero is shifted by 3 nanometers. Okay, so that means that when you are computing derivatives, first or second or third derivative, in order to search for the location of the peaks, if there were some baselines or complex baselines, the zero, or if you compute the second derivative, the minimum of the second derivative does not correspond exactly to the wavelength of the peak. It could be very drastic for some results, okay? So this is the first example. A second example is about normalization. So I think that a lot of you are using SNV or any normalization method in order to remove the multiplicative effect, which is due to some size sample or scattering or a lot of things, okay? So I have generated here a collection of spectra, very, very simple collection. So we have 3 peaks, 2 peaks here on the right. You can see my mouse, yes? On the right, you have 2 peaks, which these 2 peaks do not vary, okay? And another peak on the left, which varies with a quantity y. You can imagine a concentration of the components. Okay, so this is a good spectra, so very well measured spectra without any effect. Okay, so let's add some baselines, very simple baseline, horizontal one, translation. Okay, randomly chosen, and let's multiply by a multiplicative effect. That means that I have multiplied all the spectra with a random quantity of the shape, the form 1 plus epsilon. Let's say something a bit more than 1. Okay, so from this pure spectra, very nice spectra, I have obtained these ones. Okay, and then I have applied SNV, and these here are the results. So it's completely different, completely different from what we are searching for. We are searching for this spectra, and we are obtaining this spectra. So when you apply a model on that, there is no problem, because in fact, the information is inside. The information has been shifted from this peak to these peaks. So if you compute a multivariate model, like a PLS or classification, discrimination, what you want, PCA, what you want, you will obtain good separation, good regression, good relation with the Y, what you want. Okay, but in fact, the spectra are completely false. The variation is not at all at the good location. So we have developed recently a new method, which is an alternative to SNV. SNV means standard normal variate, and we have called this new method variable sorting for normalization. So that makes VSN, which is an alternative of SNV. Okay, so you can find the publication, the paper here, and I will present very shortly this method. So what's the problem with SNV? In fact, actually, SNV estimates the multiplicative effect as a standard deviation of the spectrum. For each spectrum, it calculates the standard deviation, and it divides this spectrum by the standard deviation in order to remove the effect of the K in the formula. And it estimates also the additive effect as the mean of the spectrum, and it removes the mean in order to remove the F of lambda, which is assumed to be a constant, okay? So it's a good idea, but these two statistics depend also on Y. If you have a part of the spectrum which is more related to Y, your compound, your variation, chemical variation, which is more related to Y than another part which is more related to the multiplicative and baseline and physical effects, you cannot process all the spectrum without taking into account these two parts. So SNV tends to dilute the information along the whole spectrum, okay? But it is what we have seen before. So one solution, a solution to overcome this problem is to calculate the standard deviation and the mean only on the wavelengths, little related to Y, and to normalize the spectrum with these values. That means that on our sample, on our example, for example, could be to calculate the mean as a standard deviation on this part, which is not related to Y, and to apply the value we have obtained on the whole spectrum. Okay, this would be the good solution. So in the real world, there is no part related only on Y and only on the physical effect. So more generally, we can think in calculating a weight related to the proportion of Y and to the proportion of physical effect, okay? And then to use this weight in the calculation of the standard deviation and in the calculation of the mean. So the problem is therefore to classify the variables according to their dependency to the shape effect, which is related to chemistry, and to the size effect, which is related to physics. So we have done an algorithm based on OneSAC. OneSAC is a very classical algorithm, classically used in image processing. And simply speaking, we draw at random a couple of spectra. And inside this couple of spectra, we draw at random two wavelengths. So we want to calculate the multiplicative effect and the additive effect, which can translate the red into the blue, the red spectrum into the blue spectrum. Because we have four values, we can compute the exact values of A and B very, very easily. And then when we have these two values, we search for all the variables, all the wavelengths which obey the same relationship. We will find the green one here. We will find that in this branch, we have more or less the same relationship between the blue and the red. And we will decide, we will call these variables the inliers. Okay, and the red one, the other variables, are called the outliers. And we repeat this operation with other wavelengths and other spectra. At the end, if we process this repetition a lot of times, we obtain a map like that with in blue the inliers and in yellow the outliers, or maybe the reverse. So we have the two classes, and we can compute the proportion for each variable. We can compute the means. That means the probability that a variable belongs to the inlier class or to the outlier class. And this probability, we take this probability in order to compute the weights. And you can see here that we will have a weight almost null for the zone where the Y is varying, and a weight almost of 1 in the zone, in the range where the Y does not vary. So if we apply this method and the weight in the calculation of SNV, you obtain the weighted SNV here with the weight here, almost 0, almost 1, and the result is here. You can see that in the part where there is no Y variation, the correction is almost perfect, and we can retrieve the peak which varies with the Y. Instead of the classical SNV, we have seen that in the classical SNV, there is some very spurious variation in this zone. So we obtain what we wanted to have. That means that we are able to calculate automatically a weight in order to calculate the mean and the standard deviation with some taking into account the two kinds of variations in the spectra. And we do not use the Y. The Y is automatically detected. There is no Y in the calculation, only the spectrum. Okay, so I will show you a real example. So the data are from a leaf, apple tree leaf taken by a perspectral imaging system. So the images are acquired with a 3 perspectral camera between 1,000 and 2,500 nanometers. And each spectrum is the mean of the pixels from an area. And we have two classes, healthy, without any fungus, and another class, which is which have been taken from spots diseased by scab, which is a fungus, a classical fungus, which attacks the apple trees. And here we have the spectra. In red, we have the scab, the disease, and in green, we have the healthy spectra. We can see without any preprocessing that the information, the main information is in this zone on the left. It is due to a difference of scattering, maybe between 100 and 104, 1,000 and 1,400. When we treat this spectra by SNV, we obtain a very clear separation between the two classes. It's very nice. And from a data learning or chemometric point of view, it's very nice because we have a very, very clear separation. But from the spectroscopist point of view, it's a bit spurious because, for example, in this zone between 140 and 1,400 and 2,000, there is a very clear separation, but there is no chemicals, there is no things that can explain this separation, okay? When we treat with VSN, we can observe that a good separation in this zone. We have seen from the naked eye that there is a very clear separation of shape, very, very different shape in this zone. But in the other zones, the difference is the separation is more subtle. And we have observed that it is really in this zone, we have a very, very difference between the shape, and also on this peak, the peak due to water absorption, that we have also separation. So this separation can be explained, this one not. So this was for the normalization. I will talk also about another innovative method we have produced last year. I think I am on time, yes, more or less. Okay, so it's quite the end. So it's about how to combine the preprocessing. So we have made a method called Sport, thanks to Federico. Thanks, Federico, for this name. It is a new way to combine pretreatments. So I will do shortly, I will explain shortly. Instead of compiling preprocessing one with, for example, doing SNV and then the SAF goals, the derivative, and then the D-trend, etc., as we can see in a lot of publications, we have thought about associating all the preprocessing in a multi-block model. That means combine in parallel a lot of preprocessing in order to take all the information which is good from all the preprocessing. So we associate the blocks with a multi-block model, which is a sequential orthogonalized PLS. I will explain very, very rapidly the functioning of this method. So if you have, for example, 4 blocks, you compute the PLS on the first one, you have the scores with L1 latent variables, you project orthogonally the remaining blocks to project this block orthogonally to this first scores, you compute a second PLS on the orthogonalized X2 block, you retain L2, LV, you orthogonalize the remaining blocks with regard to the first scores, etc., etc., etc. And at the end, you obtain a model with L1 latent variables on the first block, L2 to the second one, etc., etc. Okay? So we have applied this method on very near infrared spectra taken on mango fruits and in relation with the dry matter. So we wanted to optimize the model, and the sport has been applied just combining the raw spectra and the VSN preprocessed spectra. VSN we have seen just before. It's an alternative to SND. And what is interesting is that the best model used 4 latent variables from the raw and 7 from VSN. That means that combining the 2 information from the raw spectra and the preprocessed spectra, we are able to perform a good model. So from the raw spectra, we have the scattering because we did not process. So in the fruits, there is a lot of scattering. It is the spectra are taken in a retro back scattering, okay, in a retro diffusion. So in the raw spectra, we have a lot of information related to scattering, and in the VSN, we have removed the scattering. And what we can observe in the water region here, between 900 and 1000, we can observe completely opposite B coefficient between the 2 blocks, because with SOPLS, we can extract B coefficient for each block. And we can observe that the behavior of the model is completely opposite between the 2 blocks. And that means that that puts forward an hypothesis that the water absorption is related to dry matter. Okay, it's quite normal, negatively related to dry matter. And also that water acts on scattering. That means that there is a relationship between the water content and the scattering. It's not really surprising, but we can prove that. There is a kind of evidence in that. And this was the beginning of our hypothesis we have put forward in order to develop a new kind of model. And this research will be shown by Alexandre at the end of this session. So I invite you to stay connected until the end of this session, because you will see a new model relating the scattering and the path length to the water content. So I have finished. It is the last slide. So just for some take-home messages. So I hope I have convinced you that preprocessing are of utmost importance for chemometries. I think you all know that, but I hope that I have put the message forward that this preprocessing can have a very, very negative impact on spectroscopic interpretation. Normally, all the preprocessing we have at disposal are good for doing models, because these preprocessing have been done by chemometricians. Okay, so they are good for doing models like SNV, derivatives, etc. But you can see that the interpretation of the coefficient, of the loadings of the model, and of the spectra, of the preprocessed spectra, can be very, very altered by the preprocessing. And I have shown also, I hope that there is some active research which is underway to produce new methods that may be useful for the aquaphotomic community, because it emphasizes the interpretation of the spectra. So I thank you very much for your attention, and just I invite you to go to our web page or website in order to discover the CHEMOS research group and a lot of things about free software for chemometrics and so on. That's all. Okay.
Well, thanks, Jean-Mi, for keeping the time and for the presentation. And now we have time. You've saved a few minutes for discussion. So do you have any questions or comments? There was a comment praising the VSN method in the chat, but I don't know how. Is there, Zoltan, do you have a question? Is there a raise hand?
Yes, it's possible. Hello, everyone.
Yes, of course, do it.
Hello, everyone. Thank you very much for this concise and very educative presentation. I really enjoyed. I have a question about this VSN. We often face the problem with not only shift of the baseline, but slope problems. Does it help in this case?
Yeah. Yes, absolutely. I have presented only the very, very simple case with horizontal baseline, but in fact in VSN, we'll search automatically with the degree of polynomial and apply a weighted detrend, in fact. So you can, the actual algorithm, you can go until degree 3 or 4, okay, for the polynomials. But yes, it's exactly the same algorithm, but in place of searching for A, B, you search for A, B, G, D. It's exactly the same. Okay.
For sure, we will try. Thank you very much.
Welcome.
We have time for another short question, if any.
Well, I hope you still remember, and I hope all of you will help us with this subtraction of water spectra. You know, when bulk water is very, let's say moisture content, when you have different moisture content, then you have very close absorbance at the beginning and the end of the first overtone, like 1300 to 1600. But then you have a big difference at 1450. And there is an angle. So I want to really subtract and see the ratio between different species, water species, within this first overtone. So, and subtraction doesn't tell me anything. I mean, if I do a subtraction of raw spectra. So what we decided to do, we just decided to cut region by region, 10 nanometers or 20 nanometers, and then do the trend. So Zoltan did the trend, also on a longer region. But it's a very tedious stuff, so if it could be automatized, will be very, very nice. Okay. Because we were trying always to find very, very small difference in the spectra. Yeah, and they are very informative. Thank you.
No, you're welcome. So it's good not to do the derivative, but D20 is better. You're right.
Really. Okay, I think it's time to, we thank once again